Imagine peering through a microscope and discovering the tiny building blocks of life, cells. The study of these microscopic units led to the development of cell theory, a cornerstone of biology. All cells, regardless of their type, share a basic structure: a cell membrane, cytoplasm, and organelles. This foundation sets the stage for understanding how life operates at its most fundamental level. As we begin our journey, let's explore how these tiny structures form the basis for all living organisms, connecting us to the earliest discoveries in biology. Every cell is surrounded by a cell membrane, a dynamic barrier made of phospholipids and proteins. This membrane controls what enters and exits, maintaining the cell's internal environment. Inside, the cytoplasm serves as a medium for storage and circulation, housing various organelles that perform specialized functions. The nucleus, protected by its own membrane, contains chromatin and the nucleolus, orchestrating genetic activities. These components work together seamlessly, ensuring the cell's survival and function. Within the cytoplasm, organelles like mitochondria, ribosomes, and the endoplasmic reticulum play vital roles. Mitochondria release chemical energy in the form of ATP, powering cellular activities. Ribosomes are the sites of protein synthesis, essential for growth and repair. The endoplasmic reticulum transports substances, while the Golgi body modifies, packages, and distributes molecules. Vacuoles store materials, and their size varies between plant and animal cells. Lysosomes and centrioles are unique to animal cells, while plant cells feature a sturdy cell wall and plastids. Plant and animal cells share many similarities, but key differences set them apart. Plant cells have a rigid cell wall made of cellulose, providing shape, support, and protection. They also contain large vacuoles and plastids, which are absent in animal cells. Animal cells, on the other hand, possess lysosomes and centrioles. These distinctions reflect the specialized functions each cell type performs, contributing to the diversity of life forms. Cells come in various sizes, shapes, and structures, each adapted to perform specialized functions. This diversity allows organisms to thrive in different environments and carry out complex tasks. From nerve cells transmitting signals to muscle cells enabling movement, the specialization of cells is a testament to the adaptability and complexity of life. Understanding these differences helps us appreciate the intricate design of living systems. It's fascinating to realize that every organism, including ourselves, began as a single cell. Cell division is the process that enables growth, tissue repair, and reproduction. Through division, cells multiply, allowing organisms to develop from a single cell into complex multicellular beings. This process is fundamental to life, ensuring continuity and adaptation across generations. Mitosis is the cell division process responsible for growth and asexual reproduction. One parent cell splits into two identical daughter cells after duplicating its genetic material. These daughter cells inherit the same genetic information, except for any mutations. Mitosis ensures that tissues can grow and repair themselves, maintaining the health and stability of organisms. Meiosis is the cell division process associated with sexual reproduction. Unlike mitosis, meiosis produces 4 haploid gametes, each containing half the genetic information of the parent cell. This process introduces genetic variation, as the daughter cells are different from each other and the parent. Meiosis is crucial for diversity in populations, enabling adaptation and evolution. Cancer arises when cell division becomes uncontrolled, typically due to errors in mitosis. This unchecked growth leads to the formation of tumors and disrupts normal tissue function. Many cancer treatments target rapidly dividing cells, which is why side effects like hair loss occur during chemotherapy. Understanding cell division helps us grasp the mechanisms behind diseases and their treatments. The cell cycle is a series of phases that cells undergo to divide and replicate. It includes interphase, where the cell prepares for division, and the mitotic phase, where actual division occurs. This cycle ensures that cells grow, replicate their DNA, and produce new cells efficiently. By understanding the cell cycle, we can appreciate how life perpetuates itself at the cellular level. Rudolf Virchow, a German physician, discovered that cells divide to form new cells. This principle, known as cell division, is fundamental to biology. It allows fertilized eggs to develop into adult organisms and ensures continuity of life through both asexual and sexual reproduction. Virchow's insights paved the way for modern understanding of cellular processes. There are 2 main types of cell division: somatic and reproductive. Somatic cell division involves mitosis and cytokinesis, producing 2 identical cells for growth and repair. Reproductive cell division, or meiosis, creates gametes with half the chromosome number, enabling sexual reproduction. Each type serves distinct purposes in the life cycle of organisms. Interphase is the preparatory phase of the cell cycle, marked by high metabolic activity. During this period, the cell replicates its DNA, produces additional organelles, and synthesizes cytosolic components. Interphase is divided into 3 stages, G1S and G2. Each stage plays a unique role in preparing the cell for division, ensuring all necessary materials are ready. The G1 phase is the interval between the mitotic phase and the S phase. During G1, the cell is metabolically active, replicating most organelles and cytosolic components. Centrosomes begin to replicate, setting the stage for future division. This phase is crucial for cell growth and preparation, ensuring the cell is ready for DNA synthesis. The S phase is the period between G1 and G2, where DNA replication occurs. The cell duplicates its genetic material, ensuring each daughter cell will receive a complete set of chromosomes. This phase is essential for maintaining genetic continuity during cell division. The G2 phase follows the S phase and precedes mitosis. During G2, cell growth continues, enzymes and proteins are synthesized, and centrosome replication is completed. This phase ensures the cell is fully prepared for the upcoming division, with all necessary components in place. Mitotic division is the process by which a cell distributes two sets of chromosomes into two separate nuclei. This ensures each daughter cell receives identical genetic information. The stages of mitosis, prophase, metaphase, anaphase, and telophase, are carefully orchestrated to maintain genetic integrity. Prophase marks the beginning of mitosis. Chromatin fibers condense and shorten into chromosomes, each consisting of a pair of identical chromatids. Tubulins start to form the mitotic spindle, which will separate chromatids. The nucleolus disappears and the nuclear membrane breaks down, preparing the cell for chromosome alignment. During metaphase, microtubules align the centromeres of chromatid pairs at the center of the mitotic spindle. This region, known as the metaphase plate, ensures chromosomes are positioned for accurate separation. Proper alignment is critical for genetic stability in daughter cells. Anaphase is characterized by the splitting of centromeres, separating paired chromatids. These chromatids, now termed chromosomes, move toward opposite poles of the cell. The chromosomes appear V-shaped as their arms are dragged toward the poles, ensuring each new cell receives a complete set. Telophase is the final stage of mitosis. Identical sets of chromosomes uncoil and revert to their thread-like chromatin form. The nuclear envelope forms around each chromatin mass, nucleoli reappear, and the mitotic spindle breaks up. This stage restores the cell's internal structure, preparing for cytokinesis. Cytokinesis is the division of a cell's cytoplasm and organelles into two identical cells. It begins in late anaphase with the formation of a cleavage furrow in animal cells or cell plate in plant cells. This process completes after telophase, resulting in two separate cells ready to function independently. The sequence of events in the cell cycle can be summarized as G1 phase, S phase, G2 phase, mitosis, and cytokinesis. Each stage is essential for preparing the cell, duplicating genetic material, and ensuring proper division. This orderly progression maintains cellular health and function. Reproductive cell division occurs through meiosis, resulting from the union of two gametes produced by each parent. Meiosis reduces the chromosome number by half, producing haploid cells. This process is essential for sexual reproduction, ensuring genetic diversity and continuity across generations. Meiosis I begins after chromosomal replication is complete. It consists of 4 stages: prophase I, metaphase I, anaphase I, and telophase I. Each stage plays a distinct role in separating homologous chromosomes and introducing genetic variation through processes like synapsis and crossing over. Prophase I is the longest and most complex phase of meiosis I. Chromosomes shorten and thicken, the nuclear envelope and nucleoli disappear, and the mitotic spindle forms. Synapsis occurs, where homologous chromosomes pair off, forming tetrads. Crossing over follows, exchanging genes between chromosomes and resulting in genetic recombination. During metaphase I, homologous pairs of chromosomes align at the metaphase plate. This alignment ensures that each daughter cell will receive a unique combination of chromosomes, contributing to genetic diversity. The precise arrangement is vital for proper separation during the next phase. Anaphase I sees each homologous pair of chromosomes separate as they are pulled to opposite poles. This separation reduces the chromosome number by half, preparing the cell for the next division. The movement is orchestrated by the spindle apparatus, ensuring accuracy. Telophase I marks the end of the first meiotic division. Chromosomes are positioned at each pole, uncoil, and the nucleoli and nuclear membrane reappear. Cytokinesis occurs, resulting in 2 cells, each with half the original chromosome number. This sets the stage for meiosis II. Meiosis II follows, consisting of prophase II, metaphase II, anaphase II, and telophase II. These stages are similar to those in mitosis, but meiosis II starts with haploid cells. At the end, 4 new daughter cells are produced, each with a haploid number of chromosomes, ready for fertilization. Prophase II begins with the chromosomes in the haploid cells, preparing for another round of division. The nuclear envelope disappears, and the spindle apparatus forms. This stage sets the groundwork for the accurate separation of chromatids in the subsequent phases. In metaphase II, chromosomes align at the metaphase plate, similar to mitosis. This alignment ensures that each daughter cell will receive the correct number of chromatids. The spindle fibers attach to the centromeres, preparing for the next stage. During anaphase II, the centromeres split, and chromatids move toward opposite poles. This separation ensures that each new cell will have a single set of chromosomes, maintaining the haploid state. The movement is precise, guided by the spindle apparatus. Telophase II concludes meiosis. Chromosomes reach the poles, uncoil, and the nuclear envelope reforms. Cytokinesis follows, resulting in 4 haploid daughter cells. These cells are genetically unique, ready to participate in fertilization and contribute to genetic diversity. Meiosis introduces genetic variation through processes like crossing over and independent assortment. These mechanisms ensure that each gamete is unique, promoting diversity in offspring. Genetic variation is essential for evolution and adaptation, driving the success of populations. Cell division is vital for the perpetuation of life. It enables multicellular organisms to grow, reach adult size, replace damaged cells, and maintain a constant number of cells. Without cell division, life would not be able to adapt, repair, or reproduce, making it a fundamental process in biology. Somatic cell division involves mitosis and cytokinesis, producing two identical cells. This process replaces dead or injured cells and adds new ones during tissue growth. It ensures that the organism remains healthy and functional, maintaining stability throughout life. Reproductive cell division produces sperm or oocytes through meiosis II. This process reduces the chromosome number by half, ensuring that offspring inherit a unique combination of genetic material from each parent. It is essential for sexual reproduction and genetic diversity. Cell division ensures the continuity of life by enabling organisms to grow, repair tissues, and reproduce. It maintains a balance between cell production and cell death, keeping the total number of cells constant. This balance is crucial for the health and stability of living systems. The cell cycle is tightly regulated to ensure proper division and function. Checkpoints throughout the cycle monitor DNA integrity, cell size, and readiness for division. This regulation prevents errors and maintains genetic stability, protecting organisms from diseases like cancer. Cell division is essential for growth, allowing organisms to increase in size and complexity. From a single fertilized egg, repeated divisions produce the multitude of cells that make up tissues and organs. This process is the foundation of development, shaping every living being. When tissues are damaged, cell division steps in to replace lost or injured cells. This repair mechanism is vital for healing wounds and maintaining tissue integrity. Without cell division, recovery from injury would be impossible, highlighting its importance in health. Cell division enables reproduction, both asexual and sexual. In asexual reproduction, mitosis produces identical offspring, while meiosis in sexual reproduction creates genetically unique gametes. This dual role ensures the survival and evolution of species. In summary, cell division comes in two forms: somatic and reproductive. Somatic division, through mitosis and cytokinesis, produces identical cells for growth and repair. Reproductive division, through meiosis, creates gametes with half the chromosome number, ensuring genetic diversity. Both processes are essential for life's continuity and adaptation. Cell division is the foundation of life, enabling growth, repair, and reproduction. By understanding its mechanisms, mitosis and meiosis, we gain insight into how organisms develop, heal, and evolve. This knowledge is not only fundamental to biology, but also crucial for medical science, genetics, and the study of life itself. As we conclude, remember that every living thing owes its existence to the remarkable process of cell division.